For my TED Talk, I would like to talk about different prion diseases, and I will start off with explaining what that is. Prions are something our own body produces. They are not microorganisms, but rather proteins. The term prion refers to a misfolded protein that has the capacity to infect other healthy proteins and cause them to misfold. To get into how that happens, I'm going to have to give a brief explanation that is very, very oversimplified of how that begins to happen. In this class, we have studied DNA, so we do know that proteins basically follow and copy what the DNA does, so let's move over to RNAs. The RNA leaves the nucleus and the ribosomes will follow its coding and begin to build proteins. This is a part that pertains to folding because proteins can't function until they're folded, and they aren't necessarily folded right away. It takes a while. Proteins misfolding is a somewhat common occurrence that is typically not dangerous unless the misfolded proteins have the ability to cause other healthy proteins to misfold. Typically, the cells in our body will mark and destroy misfolded or damaged proteins, but when it comes to certain prions, our body doesn't always mark and destroy them, which allow them the ability to wreak havoc. So if a healthy protein comes in contact with a misfolded prion, the healthy protein won't be killed, destroyed, or damaged, but rather it will be infected in a way that causes it to misfold and become a prion. So what happens when a prion is able to infect a healthy protein, that newly turned prion will turn another protein into prion, and that one will do the same, so on and so forth. Nearly the same process happens when one consumes something that has been contaminated by a prion disease, but rather than your own cells being the one that creates the misfolded protein, the prion enters your system and starts spreading there. The prions will begin to spread throughout your central nervous system, eventually taking it over but remaining dormant for however long the incubation period is for that specific disease. When the incubation period is over, all the misfolded proteins will begin to clump together, and those clumps are known as amyloid plaques, and they are what cause neurodegeneration. For anyone who doesn't know, neurodegeneration is the process that causes the breakdown and decay of neurons and neural function by shortening dendritic spines, which are what cells use to communicate signals to and from one another. An example of a disease that is neurodegenerative but isn't caused by prion diseases, it would be Alzheimer's and Parkinson's disease. Now that we know the basics of what a prion is, what causes it, and how it works, let's go on to a more specific and in-depth analysis of prion diseases. Probably the most well-known prion disease would be Creutzfeldt-Jakob disease, which is what people caught when they ate meat that was infected with mad cow disease. I believe most people probably remember the panic that was going around during 2011. There's a lot to discuss about Creutzfeldt-Jakob disease, but I find that Kuru is a more interesting case of prion diseases, so I'm just going to quickly go over what would happen if one ate meat contaminated with mad cow disease. So the first thing that would happen after you consume the infected meat would be that the prions from the meat would first colonize and replicate in your secondary lymphoid organs. For anyone wondering what secondary lymphoid organs are, they are your spleen, Peyer's patches, tonsils, and lymph nodes. So, within the secondary lymphoid organs, the main site of accumulation are follicular dendritic cells, which are a type of immune cell. The prions would replicate there. Once enough had accumulated in your secondary lymphoid organs, they would then spread and reach your central nervous system through the parasympathetic and sympathetic nerves, which are the nerves connected to the secondary lymphoid organs. The passing of prions from your secondary lymphoid organs to your central nervous system is called neuroinvasion, and after that point, your prions will begin to misfold, spreading further, clump together, and then neurodegeneration will begin because the incubation period for that disease is quite small. Now time for the origin story and history of Kuru. In Papua New Guinea, the villagers of the Foray tribe had been practicing cannibalism as a funeral tradition for a long time. At one point, one of the villagers had developed a mutation in their proteins and became the first case of Kuru. When that villager died, the villagers carried on with their tradition of cannibalism and consumed the brain. The villagers that had consumed parts of the infected body became sick and infected with Kuru. So when those villagers died, the villagers would continue the practice of cannibalism, which led to more and more people being infected, getting sick, and dying from Kuru. Eventually, a scientist figured out what the connection between the villagers getting sick and their funeral tradition was, so it was explained to the villagers of the Foray tribe, and they discontinued their traditions, and from that point on, they disposed of bodies by cremation to stop any risk of further transmission. Unfortunately, many villagers still continued to die from Kuru because the incubation period of the disease was 10 to 15 years, but there were no new cases after that point, and Kuru was officially declared as eradicated sometime between 2005 and 2010. That is all, and I would like to thank you for coming and listening to my TED Talk.